So, I'm sorry that I have to break down the lecture into these small snippets right now, but I really don't trust my computer system today, so better safe than sorry. So, in the last snippet, you were informed that if there is a sufficient amount of glucose available, for instance, if you just ate, then the body will store the glycogen in the body, in the glucose in the body in the form of glycogen, and by now you know how this storage happens. Well, there is several places where it's being stored. For instance, in the muscles. So the muscle cells will contain a certain amount of glycogen and will be made available whenever these cells require glucose again. So this glycogen will be broken down. We'll come to that one later on. Apart from that, another important storage place for glycogen is the liver. Now, the liver in itself actually does not live off the glycogen being stored there and then being broken down again via glycolysis, etc., later on via glycolysis, because actually the liver metabolizes mostly fatty acids, which we'll come to later on. But the liver is the storage place to provide all the glucose required, if in any way possible, to all the cells which are not able to metabolize anything else than glucose, like, for instance, the erythrocytes and the cells of the adrenal cortex, that is the Nebennierenrinde. But let's take one step after the other. So now the cells picked up a sufficient amount of glucose so they could afford to store a bit of the glucose in the form of glycogen. And later on, maybe because the organism has to strain itself for whatever reason, maybe you're doing sports, I really don't know. So somewhere later on, long after you had your last meal, sorry, this was not religious content, of course, so the last time you ate something, glucose is required again. So that's exactly what you stored the glucose for, in the form of glycogen. So this glycogen is being broken down to, in the end, deliver glucose again, which then can undergo glycolysis and make sure that the organism gets all the energy required. The idea here is basically you've got your strand of glucose molecules forming the glycogen with all these side branches, etc. And the glycogen is being broken down by simple phosphate ions to deliver glucose 1-phosphate again. So the glucose 1-phosphate you've already seen. But it's quite an important thing that this reaction, where the individual monomers of the glucose and the glycogen strand are being cut off one after the other, is not a hydrolysis, even though it formally looks like it, but in fact it's a phosphorylysis, because the attacking agent is not a water molecule, which would be aqua, would be or hydro, so hydrolysis. It really is the phosphate ion which goes to the ultimate, the final, the terminal glucose molecule of a glycogen, and then breaks this alpha-1,4 glycosidic bond to the penultimate glucose here, and cleaves it by forming the glucose 1-phosphate. The rest of the glycogen chain just remains unaltered, except that one glucose has been broken off. This is not a reaction where ATP is involved, neither as the one facilitating the whole reaction by delivering the energy. The phosphorylysis taking place here can be done by the phosphate ions themselves. And there is also no ATP being produced in this process. Not yet. That's what's going to happen later on when the glucose 1-phosphate is metabolized any further, probably undergoing glycolysis, so that in the end ATP is synthesized. Of course, once again, as almost all biochemical reactions, this reaction once again has an enzyme. It's the glycogen phosphorylase. Hello. So, once we've got this glucose 1-phosphate, which is just a normal ester of the phosphoric acid with glucose at the hydroxyl group of carbon number 1. This is being isomerized to glucose 6-phosphate, in brief G6P. Well, you know that one, not only from glycolysis, but also because in the last video session I've told you that this is the actual form in which glucose is present in the bloodstream, in the cells, in the whatever. In most cases, there's hardly any free glucose. In most cases, it's glucose 6-phosphate, which of course is also just an ester of the alcohol glucose with the acid phosphoric acid. So please do not mix those up. Do not confuse those with some of the energy transfer molecules you know from the penultimate video session, where I've shown to you that occasionally we're talking about phosphate esters and occasionally we're talking about mixed anhydrides. Both glucose 1-phosphate and glucose 6-phosphate are just esters. So they're both not really high energy molecules. Now, to completely break down our glycogen, we also have to break down the side chain elements which are connected alpha-1,4. And in the end, we even have to degrade the 1-glucose rest, which is attached to the main strand of our glycogen via an alpha-1,6 linkage. And this requires a cofactor. It's pyridoxal phosphate. We'll talk about that several times, once after this slide here, and you will re-encounter it again and again in the weeks we've got to come. So, this slide here probably looks rather similar to one you've seen in the last video session. Now here, let's have a look at how this debranching works. The first step almost looks as if it were the reversal of the formation of glycogen, where, as you might remember, an already existing longer strand of alpha-1,4 connected glucose units were taken from one strand, in most cases from a side branch, to an individual to form a new alpha-1,6 bond so that another side branching could be formed, like you can see here. Now, breaking down our enzyme, not our enzyme, there is no enzyme being broken down. So now breaking down our glycogen, the first thing is to minimize the branching. So you'll take a part of a branch here, of a side branch, actually 3 glucose units, which are being cut off, once again using an enzyme which, lovingly is called glycogen debranching enzyme, aren't they creative in their names, to reintegrate this part, these 3 glucose units, into the main strand, which then can once again be cut off one after the other, after the other, after the other, just like you've already seen it, which only now leads to one alpha-1,6 connected glucose side chain thingy without anything else being attached to it. And this alpha-1,6 glycosidic bond can be cut by or broken by the debranching enzyme again. So basically the breaking down of glycogen to glucose consists of A, nibbling off the main strand one after the other, and B, if necessary, if there is a side chain, to take a small snippet from it, 3 glucose units, and reintegrate it into the main strand, which then once again is being nibbled off one after the other. Now, you might remember, because it's about 2 minutes ago, that I've told you that this breaking down of glycogen requires the cofactor pyridoxine, which is also called vitamin B6. And once again, you can see it's a relatively simple molecule. 1 methyl group, 1 hydroxy group, and 2 hydroxymethylene groups. Now, actually, this is not the pyridoxal phosphate, which is the actual cofactor, but you will get to see both the pyridoxine and the pyridoxal phosphate again and again in biochemistry, because It's an important cofactor in the metabolism not only of glucose in itself and the breaking down of glycogen to glucose, but also in the metabolism of amino acids. In other words, this pyridoxal phosphate is another of these wonder molecules allowing us to play Lego even in biochemistry, but one after the other. Please compare pyridoxine, which has in para position or in 4 position to this hetero atom, the nitrogen in this system here. There is a primary alcohol, CH2OH. Now, this primary alcohol in pyridoxal phosphate is already oxidized to the aldehyde. And, going back again, the other primary alcohol here at position number 1, 2, 3, 4, 5 of our pyridine ring, which is also a primary alcohol, this primary alcohol is being esterified with phosphoric acid again. So it's phosphorylated. So it's not phosphorus related, it's phosphorylated. Please do not mix up these two terms. And you will see that this phosphorylation of one or the other alcohol group plays an important role in biochemistry, even up to the point I'm not even touching with a 10-foot pole, which is genetics and epigenetics. Their phosphorylation obviously plays a vital role as well. But I'm not going to delve into genetics here. So this is our pyridoxal phosphate, definitely the most important cofactor in the metabolism of amino acids, and as you've seen in breaking down of glycogen. And without wanting to go into too much detail with respect to the metabolism of amino acids, because I intend to give an individual lecture on that one slightly later on, but just to show you what this stuff is required for, there is something we've already dealt with when we talked about amino acid quite a few lectures ago, where we had the transfer of one amino group from one molecule to another. So for instance, if you take the aspartic acid or the anion, the aspartate, please add some E's for good measures here because this used to be a German slide. So, and you compare that with the alpha-ketoglutarate acid or alpha-ketoglutarate, another molecule you've already seen if my memory serves me well, and you will definitely re-encounter when we talk about the citric acid cycle. Now, basically what's happening here is not only playing Lego, it's actually switching partners. The carbon bearing the amino group of our aspartate under the influence of the enzyme of an aminotransferase actually is not only being brought to the amino group, is not only brought to the reaction partner, this is the ketoglutarate, but under the same circumstances, the carbon bearing the amino group is being oxidized. You remember in biochemistry, oxidation was gain of oxygen or loss of hydrogen. This one here both gains oxygen and loses hydrogen. So if this is not a carbon which is to be oxidized in the end, then I really don't know which one is. And obviously this carbonyl group of the alpha-ketoglutarate is being reduced. The oxygen gets lost, instead there is a nitrogen attached to it, and the carbon in the end has a hydrogen. So in other words, amino group plus hydrogen are exchanged for a doubly bound oxygen, or vice versa. A doubly bound oxygen is exchanged for amino group plus hydrogen. And this transfer of just one amino group, this oxidative transfer or this rejective transfer, is a typical step in biochemistry, and that's why I've been pestering you with with the names and the structures of so many of the biomolecules required for biochemistry, because they really occur again and again. You already know the oxalic acetate, or oxaloacetate in English, and you will re-encounter it today. And of course you already know glutamate. This is another of the proteinogenic amino acids, as is the aspartate, of course. Okay, so how this pyridoxal phosphate, in brief pulp, PALP, does that is something you will probably get to know slightly later on. At the moment, it's just important for you to know that pyridoxal phosphate is able to do something like that, and it's obviously important in biochemistry. Okay. And now we've seen that our glycogen actually can be formed from glucose, but it's also possible for the glycogen to be broken down to form glucose again. And this is being facilitated, of course, under the influence of enzymes. And as we've got two reactions here, which actually are directly working against each other, it obviously becomes extremely important that these things do not happen uncontrolledly in any way. Now, it stands to reason that in a situation where, due to food or other circumstances, the glucose level within the cells is high enough, then that means there is enough glucose to provide for the energy, to use some of the energy to, provide for the energy required and use some of the energy to store the glucose excess in the form of glycogen. If, on the other hand, glucose is required for several reasons, then it stands to reason that the glucose should be no problem at all for the organism to release glucose from the glycogen again. Now let's have a look at what happens here, and to do that we first have to know that the enzyme, the phosphorylase, the one we use for the oligomerization or the polymerization of glucose to glycogen, actually can exist in, depending on what textbooks you ask, you look it up in, 2 or 4 different forms. And both answers are correct, but first things first. So, as with many other enzymes, for the phosphorylase, there is an active form, that is the relaxed form called the R form, where the enzyme actually does what it's supposed to do. And there is a T form, the tense form, where there is a certain tension in the system and it's inactive. Okay, so 2 forms, the R form, the T form. You might already know that from hemoglobin. And if you don't, it really doesn't matter right now. So, but these 2 forms can be converted into one another. Now, it's important to know that it is the R form which has the activity of saying, okay, there is some glycogen present and I might need it right now, so I will start breaking down glycogen, well, moderately, wherever if it's required at the point in this system, wherever this phosphorylase might be. So this is really something happening in almost all the cells. Just because I mentioned the muscle cells and the liver does not mean that these things are happening exclusively in those 2. Well, assume for a moment we're within the muscle. Then that means if some glucose is required, because in the end we want to have glycolysis and then get some ATP, then this phosphorylase will undergo this reaction. Moderately, but it will do so. Now, this is being controlled by the level of both ATP, which is of course the energy deliverer per se. You know, this was our energy currency. So in other words, if there is a sufficient amount of ATP, then this phosphorylase says, actually, at the moment, I don't have to work, I don't have to break down glucose, because you would only use the glucose to make ATP, and obviously we've got enough of that. So the active R form, the relaxed form, in the presence of ATP, is being converted into the inactive T form. Now, this is a typical case of allosteric control. The same allosteric control works with glucose 6-phosphate because this is the first product of the glycolysis, and it's the first product, it's the isomerization product of the breaking down of glycogen anyway. So in a way you could say this is product controlled. If there is already the product of the reaction present there, Le Chatelier comes into play and says, well, there's enough product, why should I let this reaction continue? If on the other hand these cells contain a relatively large amount of AMP, so adenosine monophosphate, then this basically can be considered as a signal for the cell to say, well, if the level of AMP is so high, that means that my ATP level must be threateningly low. I guess it's time for me to work and break down a little bit of glycogen to get glucose so that it can undergo glycolysis, and then ATP will be restored and the AMP levels will be lowered. So far, so good. That's what happens in the everyday life when the organism is not under a major strain. This is just everyday existence. If, on the other hand, so this would be the control of breaking down of glycogen just via allosteric control. So far, so good. Now the next step. If the system is under stress, and in this case with the system I really mean the organism, like for instance an animal in fight or flight situation. You might know that, and even humans occasionally encounter that. If the whole system basically says this is definitely a situation where I'm going to need lots of energy very fast because either I have to run away or I'll have to give a fight, then the ATP present already is being used to phosphorylate our phosphorylase enzyme. For those really interested in proteins, yes, of course, we know the primary structure, that is the series of amino acids making up the phosphorylase, and it's the serine at the position number 14, so the 14th amino acid from the N-terminus is a serine, and serine, you know, has got a hydroxy group, so this hydroxy group is phosphorylated. Once again, it's just a small modification, but we're talking about a covalent bond, so this is a covalent modification here. And now we've got the still inactive T form of our phosphorylase, which now is phosphorylated. Yes, try saying phosphorylated phosphorylase 3 times, but I bet you once again this really brings lots of points in Scrabble. Okay, so this phosphorylated phosphorylase now gets a slightly different name. The normal everyday phosphorylase responsible for doing what you do in an everyday life with the moderate activity is called the phosphorylase B. This is the non-phosphorylated version, and the phosphorylated version then is called phosphorylase A. And this stuff facilitates the degradation of glycogen in a very, very short time. So the moment that the system is under stress, like I said, like in the fight or flight situation, then this allows the organism to break down glycogen at a far, far higher level than before, so that far more glucose, and as such glucose 1-phosphate is being produced, and then glucose 6-phosphate, and then you can undergo the glycolysis so that really tremendous amounts of energy can be released at a very short time. Which of course only makes sense if there is not already a sufficient amount of glucose present, not even under a stress situation. So once again, even the activity of this phosphorylase A, which once again was the covalently modified version with 2 phosphates attached to it because the enzyme itself is the homodimer. So the activity of this one is hindered if the level of glucose in the cell is high enough. That's what this red stands for. This leads to the reversal of the reaction. If the glucose level is too low, then the phosphorylase A will go into the active R form and then start to phosphorylate glucoses like hell, so that glycogen terminals like hell, so that in the end you get lots of your individual phosphorylated glucoses. But if the level of glucose is high enough, then this will be stopped again. So once again, this is a strictly controlled metabolistic path. And there is even more, because this is once again just the tip of the iceberg. I've just told you something about how to control whether or not glycogen is being broken down. The next step is, how is the synthesis of glycogen, the formation of glycogen from individual glucose directly after food intake or the like being controlled. Now there is another enzyme, we've got that one. Then there is calcium ions playing an important role, and the whole thing is controlled by hormones as well. So even though this in itself is, chemically speaking, relatively easy to understand, I stress the relatively intentionally here, but if you want to look at the whole thing, then you will see that we'll talk about 10 to 12 pages of a decent textbook, how the actual glycogen metabolism is controlled beyond this one slide here. And I think you will notice that I've talked quite a bit about just this one slide already. Okay, the next step, because I really don't want to go into that much detail. If you want to look it up, there are several brilliant textbooks on biochemistry and with respect to the regulation of various enzymes, often up to the hormonal level, which of course is highly interesting, but you have to know quite a bit about hormones, and I didn't even start that topic yet, except for mentioning that some proteins also work as hormones. So this will go far beyond the scope of this lecture here at the moment. But for technical reasons, once again, I will cut this lecture short here. The next step would be the synthesis of glucose from something else. That is the de novo formation of glucose, called gluconeogenesis. We'll come to that one soon.